Hi! This afternoon, magre-review tayo on lipids. So, lecture style siya. Wala muna akong masyadong questions. Although, may mga 1 or 2 questions every now and then. So, review lang ng lipids and then pupunta tayo on isolation of lipids, qualitative test, and thin layer chromatography. So lipids is the fourth class of biomolecules in this course. So napag-aralan nyo na proteins, nucleic acids, and then last week nag-review tayo on carbohydrates. So ang pinagkaiba ng lipids, unlike the former 3 biomolecules, ang lipids natin, they are not polymers. So 'di ba yung proteins, polymers sila ng amino acids, nucleic acids, polymer ng nucleotides. And then carbohydrates, polymer sila ng monosaccharides. So lipids are not polymeric, but their special characteristic is that they aggregate. So they can form structures like this, like the liposome, the micelle, or a bilayer. We are all familiar with the bilayer. This is the structure of your cell membrane, right? But for example, the liposome, one of its famous applications is in drug delivery. So gumagamit sila ng mga lipids, pinapa-assemble nila into liposomes, tapos yung drug nandun siya sa loob ng liposome. And then they target the liposome to a specific organ, for example, to treat a disease. Compared to other biomolecules that you have studied, lipids, in terms of structure, they are more diverse. So when we say diverse, mas marami tayong makikitang itsura ng mga lipids. Unlike your proteins, ang proteins, di ba, meron ka lang peptide bonds linking your amino acids. Tapos yung amino acid natin, meron kang central carbon, meron siyang side chain, may amino, may carboxyl functional groups. Si nucleotides, repeating lang siya ng sugar, nitrogenous base, phosphate. Tapos yung carbohydrates natin, repeating monosaccharides lang siya. Now yung lipids, as you will see or probably sa lecture nakita nyo na, mas diverse yung structures nila. And the only thing they have in common is their property, hydrophobicity. So they are not soluble in water, which is taken advantage of when you try to purify or isolate lipids. So the primary function of your lipids are 2. Okay? The first primary function or major function, yung sikat na function nila ay components of biological membranes. And then the second is energy storage. But your lipids, their functions are not limited to these 2. So may iba, very minor and uncommon actually, but still important. Lipids can also act as cofactors for your enzymes. Some can also act as electron carriers, light-absorbing pigments, hydrophobic factors for proteins. Some lipids even act as chaperones to help some membrane proteins fold. Some are emulsifying agents, some act as hormones, and some are involved in intracellular messaging. So the first class of lipids that one studies in biochemistry is your fatty acids. So the fatty acids are the simplest. So they are just hydrocarbons. Mainly, they are alkyl chains. So may alkyl chain ka, tapos sa dulo niya meron siyang carboxyl group. So these hydrocarbon derivatives are in their most reduced state. So when you say reduced state, wala kang makikitang double bonds masyado, wala ka rin makikitang functional groups na may oxygen. So if you look at the structures, for example, of stearic acid. So this is stearic acid composed of 18 carbons. You see that you have a very long hydrocarbon chain, and then you only have one functional group at one end, which is your carboxyl functional group. So, reduced siya. Well, it's not completely reduced because you still have the carboxyl. But compared to other biomolecules, for example, yung carbohydrates marami siyang OH groups, di ba? Your fatty acids are more reduced, in a more reduced state. So fatty acids can have 4 to 36 carbons. So they can be as short as your ano nga ba tawag kapag 4 carbon na carboxylic acid? Butanoic acid. Ayan. So that's the simplest fatty acid, up to 36 carbons. Pero yung pinaka-common na fatty acids would be from 12 carbons to 20 carbons or 22. So these fatty acids are common components of another class of lipids. We call them triacylglycerols, which we will see later. So here we see some of your biological fatty acids, starting from 12 carbon fatty acid, lauric acid, 14 carbon myristic acid, palmitic acid, stearic acid, and so on. Now you have two classes of fatty acids here, saturated and unsaturated fatty acids. So when we say saturated, all of your bonds in the hydrocarbon chain are single bonds, like stearic acid. So stearic acid is an example of a saturated fatty acid. And when we say unsaturated, it means the hydrocarbon chain has at least one double bond. So pag may isa kang double bond, unsaturated na yon. Example, oleic acid has a double bond between carbons 9 and 10. Yan. So in higher plants and animals, the predominant fatty acids contain 16 carbons to 18 carbons. So nandun siya sa gitna ng range natin. So 'di ba kanina sabi, fatty acids can contain 4 carbons to up to 36 carbons. So yung pinaka-common nandun sa gitna ng range. Fatty acids containing less than 14 carbons and greater than 20 carbons are not very common. Now question, why do most fatty acids contain even number of carbons? So if we go back to the table, wala tayong makikitang odd-numbered fatty acid. So bakit lahat sila even-numbered? Even yung number ng carbons. Anyone? Sino may idea? Di ba nasa ano na kayo? Nasa metabolism na kayo? Ang sagot ay kung paano sinisynthesize ang fatty acids. Anyone? Janelle. Nagtawag. Janelle. Bakit even lang ang number ng carbon? Hello?
Hello po.
Ayan, okay.
Ma'am, rinig po ako.
Yes.
Kasi po, kapag nagsisynthesize daw po ng fatty acids, yung carbon acetyl CoA molecules daw po yung na-assemble together. Yung length daw po nun ay by 2 carbons each.
Okay.
Yun po. Thank you po.
Thank you. Very good. So, yung synthesis ng fatty acids kasi, yung introduce ng acetyl-CoA, di ba yung acetyl-CoA, 2 carbon siya, tulad ng sabi ni Janelle. So, every acetyl-CoA kasi, so binibuild niya yung fatty acid chain mo by 2s. Kaya, ang nakikita lang natin ay even-numbered fatty acids. Ayan. And then, when you name fatty acid, so nakita natin kanina dun sa table, nakita actually natin, di ba, yung sa table, meron siya ditong 12:0, and so on for your saturated fatty acids. For your unsaturated fatty acids, you have, for example, kay oleic acid, 18:1. So this is actually a system for us to identify your fatty acids. So yung first number, it tells you the number of carbon atoms. And then, the second number tells you the number of double bonds. So pag nakita nyo, 18:0, ibig sabihin saturated fatty acid siya. Wala siyang double bond. And the way you indicate the position of your double bond is by, so you enclose in a parenthesis your delta, tapos may superscript siya. This number tells you the position of your double bond when you number the carbons starting from the carboxyl functional group. So you start from this carbon, you number this one. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So yung head ng double bond natin is at carbon 9. So yung superscript ng delta is 9. Okay, so this is your oleic acid. Tama? This one. Oleic acid. So 18 carbons with one double bond at carbon 9. Yan. And then, another example is this one. So by looking at this symbol, we know that the fatty acid has 20 carbons and it has 5 double bonds. So nasaan yung mga double bonds niya? So carbons 5, 8, 11, 14, and 17. So you can easily draw the structure of this fatty acid. So mag-drawing kayo ng carboxylate and then draw 20 carbons. Tas may double bond ka from between carbons 5 and 6, carbons 8 and 9, carbons 11 and 12, carbons 14 and 15, and carbon 17 and 18. Now, another method that they use to classify your fatty acids, label and classify your fatty acids is this omega system. So, yung omega, ina-assign na omega yung pinakahuling carbon. Yung pinakamalayong carbon from the carboxyl functional group. So, this is your omega. And then, you assign your fatty acid whether it is an omega-3, an omega-6, an omega-9 fatty acid, depending on the position of the very first double bond from the omega carbon. So for example, this one, omega-3 fatty acid siya. Kasi yung pinakaunang double bond from the omega carbon is at carbon 3. So bakit may ganong classification? Kasi napansin nila na yung double bonds, medyo fixed kung saan sila nag-occur dun sa hydrocarbon chains ng fatty acids. So, medyo maraming omega-3 fatty acids and omega-6 fatty acids. So, this one, eicosapentaenoic acid is an example of an omega-3 fatty acid. Ayan. So, for example, these four fatty acids, si stearic acid, we write its symbol as 18:0. Tapos si oleic acid, 18:1, delta 9. Linoleic acid, 18:2, delta 9, 12. And si alpha-linolenic acid, 18:3, delta 9, 12, 15. right? Si alpha-linolenic acid is an example of an omega-3 fatty acid, right? So this is your omega carbon. Yung pinakaunang double bond from the omega is at position 3. So it's an omega-3. Pero si linoleic acid, this is an omega-6 fatty acid. This is your omega carbon. And then yung position ng pinakauna from the omega, pinakaunang double bond, is at 1, 2, 3, 4, 5, 6. So this is an omega-6 fatty acid. Si oleic, 1, 2, 3, 4, 5, 6, 7, 8, 9. It's an omega-9 fatty acid. So that's how you use this system in naming your fatty acids. Hindi siya exactly name, but it's a way to label your fatty acids. So in this table, you will see some properties, the melting point, specifically the melting points of some fatty acids and their solubility in water and benzene. So notice, yung solubility, sobrang baba ng solubility in water, but they have very high solubilities in benzene. But I want you to take note of the melting points. What can you say about the melting points of your saturated fatty acids and unsaturated fatty acids? So ito yung, okay, above this line, you have saturated fatty acids, and then below the line, you have unsaturated fatty acids. So anong napapansin nyo? Anyone? How about si kalimutan ko nickname mo. Si Jan. Kahit sa chat lang sumagot. Ayan, si John Lu. Anong pansin nyo? Baka may ano yung audio ni John Lu. Kita naririnig.
Parang hindi nga po marinig, ma'am. Pero yung sa... Ay, ayun po, nag-chat na lang po si John Lipat.
Hindi ka rinig. Try mo mag-leave audio tapos balik ka ulit. Connect to audio ka ulit. Mas mababa yung melting points ng unsaturated fatty acids. Tama. So, tingnan nyo yung melting points nung may double bonds. Yung iba nasa negative na, di ba? Pero, yung saturated, lahat sila above room temperature. And, isa pa, habang humahaba yung hydrocarbon chain, tumataas din yung melting point. So, ang butter, for example, ano kaya yung karamihan ng fatty acids na nasa butter? Saturated o unsaturated fatty acids? Butter, lard. Anong type ng fatty acids yung nakikita sa butter? Lard or ano pa ba? Margarine. Vegetable shortening. Saturated or unsaturated?
Ma'am, saturated po.
Dinig na si John Lu. Ayan. Saturated. Okay, so lahat ng anything fat na may fats, tapos solid at room temperature, saturated fatty acids, most likely. Kasi yung unsaturated fatty acids natin, negative yung melting points nila eh. So, ibig sabihin, at room temperature, liquid na sila. Like your cooking oils. Diba? So, bakit ganon? Yung behavior nila. So, it's because, wag mo na yan. It's because of the structure of your fatty acids, yung molecules. So yung saturated fatty acids, even though each carbon-carbon bond has the freedom to rotate, so di ba sa organic chemistry, alam nyo na, pwedeng umikot yung carbon-carbon bond nyo. May freedom lahat ng carbon-carbon bonds na mag-rotate. Pero yung pinaka-stable na form nila is kapag diretso yung hydrocarbon chain. So ganito siya, diretso siyang ganyan. Kasi pwedeng umikot, for example, itong carbon-carbon bond na to. Iikot siya ng konti, so hindi na siya diretso, di ba? Pero diretso yung pinaka-stable na itsura ng molecule. Pag unsaturated, so meron kang double bond. And ang fatty acids natin, kapag may double bond sila, usually cis yan. So when we say cis, yung ano tawag dito, substituents on both sides ng double bond, nakaharap sila sa parehong side. So just like this one, so itong substituent na to, itong buong chain na to, diba? Parang nandun siya sa left side ng double bond. Likewise, this substituent nandun din sa left side ng double bond. Kaya cis double bond siya. They are both on the same side. Pag trans, meron din tayong tinatawag na trans double bond. Then, itong chain na to, nandito siya. Nasa kabila siya. So, diretso. But again, most of your fatty acids tend to have a cis double bond. And this cis double bond introduces a bend in your hydrocarbon chain. So dahil doon sa bend na yon, kapag nagdikit-dikit na yung fatty acids nyo na unsaturated, hindi maayos yung arrangement nila. So there are spaces in between them. So we talk about packing of your molecules. So they cannot efficiently pack. Therefore, hindi nila kayang mag-solidify even at room temperature. So bakit hindi nila kayang mag-solidify? So because of the spaces, they don't feel the intermolecular forces of attraction that much. Kasi nga hindi efficient, hindi sila dikit-dikit. So liquid sila at room temperature compared to your saturated fatty acids. So because all of them are assuming this straight hydrocarbon chain orientation, they can pack very tightly. They feel the intermolecular forces of attraction more than in this state. Kaya solid sila at room temperature. Okay? So that explains the property of your unsaturated and saturated fatty acids. Now, for your saturated fatty acids, I mentioned that as the hydrocarbon chain lengthens, the melting point also increases. It's because, why? Sino may idea? Bakit pag humahaba yung hydrocarbon chain ni fatty acid, tumataas yung melting point niya? Ni Juan?
Kasi po, hydrophobic po yung carbon chain length, ma'am. Kaya pag mas humahaba po, mas nagiging insoluble po.
Lahat sila insoluble. So, ang nangyayari is, in fact, pag tinignan nyo yung solubility at benzene, habang humahaba, nagiging less yung solubility niya, 'di ba? So, hydrophobicity doesn't exactly explain the behavior of the melting point. It's more of yung strength of van der Waals forces. Ay, may sasagot dapat. May sasabihin.
Hindi po ma'am, napindot ko lang po.
Okay. Yung strength ng van der Waals forces. So, pag meron kang non-polar molecule, kasi di ba meron tayong polar, dipole, ayun, dipole-dipole interaction, pag polar yung molecules. Pag non-polar sila, so since wala kang dipole-dipole interactions, meron lang silang van der Waals. Na yung strength ng van der Waals, nagde-depend siya kung gaano kalaki yung molecule mo. Pag mas malaking molecule, mas maraming electrons, mas malakas yung van der Waals forces na kaya niyang i-exert sa isa pang molecule. So, mas malakas yung van der Waals, mas attracted sila, mas malakas yung force na nag-hold sa kanila, kaya mas mataas yung melting point. So, kailangan mo ng mas maraming energy para ma-melt siya. Kasi mas malakas yung interaction ng molecules habang lumalaki sila. That is 4 van der Waals forces. Okay? And then, ito palang slide na to, nakalimutan ko siya. So, i-mention ko dapat dito yung omega-3 fatty acid. So, naririnig nyo siguro yung omega-3 fatty acid sa commercial ng mga canned tunas, ganyan. So, yung omega-3 fatty acid kasi, sila yung healthy fatty acids. And then, meron din iba na, syempre, hindi healthy. And nire-recommend, actually, merong recommendation, minabasa ko na yung recommended ratio ng omega-6 to omega-3 fatty acids sa diet is 1:1 to 1:4. So dapat mas marami, if not 50-50, dapat dapat if not 50-50, mas marami yung omega-3 fatty acids. So 'di ba meron din mga supplements ng ng mga fish oil ganyan, rich in omega-3 fatty acids. Ayun. So kasi omega-3 fatty acids daw are the healthy fatty acids. Tama ba? Yeah. However, so meron sinabi din na yung diet daw, well, this is in America, yung common diet nila, ang ratio 10:1 to 30:1. So, mas marami yung omega-6 fatty acids, which is not really good kasi nga yung ibang fatty acids, they are associated with the occurrence of cardiovascular diseases. So yung mga omega-3 fatty acids, aside from fish oil, meron din sila sa leafy vegetables. Ayan. Okay. So that is for your fatty acids. So the next class of lipids is your triacylglycerols. So the triacylglycerols are the simplest lipids made of fatty acids. So yung structure niya, it requires a glycerol molecule. So this is your glycerol molecule. And then you have 3 fatty acids condensing with this glycerol molecule. So meron kang, ito yung original na glycerol, right? And then you have esters of your fatty acids. Okay? Ayan. So yung OH, napalitan siya ng fatty acid. So the simplest that you can create is glycerol plus one type of fatty acid. So ibig sabihin, tatlo, for example, pag sinabing tripalmitin, meron kang tatlong palmitic acids na magko-condense with your glycerol. Or pag sinabing tristearine, then you have 3 stearic acids condensing with one glycerol molecule. Pag sinabing triolane, you have 3 molecules of oleic acid condensing with one glycerol molecule. Pero, hindi yun common. Ang mas common is magkakaiba yung fatty acids na kumakabit sa glycerol. So this is an example. So you have 1 palmitoyl, 2 linoleol, 3 stearoyl glycerol. So this is an example of a triacylglycerol, other name, triglycerides, fats, so ito yung fats, and then neutral fats. Neutral because yung fatty acids natin, originally charged dapat yon, di ba? So deprotonated yung carboxyl functional group at pH 7. But since it condenses with your glycerol, nawawala na yung charge na yon. That is why your triacylglycerols are called neutral fats. So wala silang charge. So they are completely hydrophobic, completely non-polar. And their main function is the energy storage. So yung mga fat deposits sa ilalim ng balat natin, dito sa belly area, puro mga triacylglycerols yon. And thus, they are the most abundant type of lipids in animals. So here you see a picture of cells with fat deposits inside. These are animal cells, yung nasa letter A. So yung kulay puti na yan yung fat deposits. Tapos kung meron kayong nakikitang red spots, those are the nucleus. So this one are probably what we call adipocytes or your fat cells. Cells which are completely dedicated to storing fats or your triacylglycerols. In plants, there are also cells in plants that also store your triacylglycerols. So dito, yung nakikita niyong puti sa labas niyang dark spheres na yan, those are the fat deposits. Okay. Trans fats. So, yung trans fats, I'm sure narinig nyo na rin yung trans fats. This one are, okay, yung trans fats, nagkakaroon ka ng trans fats through the process of hydrogenation. Okay, so meron tayong tinatawag na hydrogenation and is usually done to improve the stability of cooking oils. So yung ibang cooking oils, para mas ma-withstand nila yung mataas na temperature, nagkakaroon ng hydrogenation or partial hydrogenation. Ang aim kasi doon is, most of the fatty acids in cooking oil, initially, may mga double bonds sila. Now, para, di ba, napansin natin yung unsaturated fatty acids, mababa yung melting point. Pag tinaasan pa natin yung temperature nila, chances are, yung structure nung unsaturated fatty acid mo, masisira na. Like, magbe-breakdown na sila into smaller molecules. Now, if we increase the melting point, kung patataasin natin yung melting point, even just slightly, that helps in the stability. Kasi ang mangyayari, since mas mataas relatively yung melting point niya, it means that yung energy, kapag ininit mo yung oil yung energy na yon gagamitin mo na niya para ma-overcome yung intermolecular forces of attraction, para mapaghiwa-hiwalay mo muna yung molecules. Kasi kung mababa yung melting point niya, ibig sabihin, relatively, magkakahiwalay na sila. So pag ininit mo siya, yung heat ginagamit na para masira yung bawat molecule. So they perform partial hydrogenation. So ang effect nun is yung ibang double bonds, ginagawa nilang single bonds. So from the name itself, hydrogenation. So yung double bonds, instead na double bond siya, magkakaroon ng hydrogen. So meron kang additional single bond for each carbon. However, yung side effect ng partial hydrogenation, nagkakaroon ka ng trans fats. And trans fats is also related or linked to cardiovascular diseases. And pinapataas din daw niya yung proportion ng low density lipoprotein, which is your bad cholesterol. So, ayan, kaya ako may slide on trans fats. Yan, doon sa reference ko, kinuwento niya yung kung paano nagkakaroon ng trans fats. So, pag gumamit ka ng, actually, hindi lang siya limited sa cooking oils eh. Even sa processed food, or kapag nag-bake ka, for example, ganyan. So, meron kayong makikita mga ibang commercial products aside from cooking oils na dun sa label nila nakalagay, no trans fats. Yon. Okay. And then, I forgot to mention, for your triacylglycerols, aside from energy storage, another function is insulation for animals especially. Ayan. Aside from fatty acids and triacylglycerols, we also have biological waxes. So these are formed by, you have a fatty acid and a long chain alcohol. So these two molecules will form an ester. This is an example. So you have a palmitic acid here and 30 carbon alcohol, which is called triacontanol. Yan. So magko-condense sila, forming an ester. And this one is the major component of beeswax. Ayan. So these biological waxes, their primary function is for water repellent. There are plants that produce these biological waxes to prevent water evaporation, for example. So kung meron kayong napapansin ng mga dahon na parang oily yung surface nila, most likely biological wax yon. So ester yon. And yun yung nakakatulong para hindi masyadong mag-evaporate yung water from the leaves. Aside from that, these waxes are actually the primary energy storage of planktons. Ayan. So yung energy storage, hindi as a function, hindi lang siya limited to your triacylglycerols. So planktons reserve their energy through these waxes. Okay. So, naaral na natin fatty acids. Na-review na natin yung fatty acids, triacylglycerols, konting mention about your waxes. Let's go to your structural lipids. So, yung structural lipids natin, here's a chart showing the types of particularly membrane lipids that you will encounter. So, you have phospholipids, glycolipids, and also some lipids found in archaea. So ang focus natin ay ito. Glycerophospholipids, sphingolipids, and your galactolipids and sulfolipids. So your glycerophospholipids, or simply called phosphoglycerides or phospholipids. The main structure is, again, you have your glycerol molecule. So, balikan natin si glycerol from your triacylglycerol. So, this is your glycerol. The difference is that you have a phosphate group attached to carbon 3 of your glycerol. Okay? And then, for carbons 1 and 2, meron kang fatty acids na nakakabit sa kanila. So, ang pinagkaiba niya sa triacylglycerol, si phospholipid, phosphate group ang nakakabit sa carbon 3. So, unlike your triacylglycerol, which is a neutral fat, your glycerophospholipids or simply phospholipids are charged. So, they have a polar head, a charged polar head, and then the rest of your the structure of your phospholipid are non-polar kasi ito na yung hydrocarbon chains natin. So here are some types or examples of phospholipids. So nagdedepende sila kung ano yung nakakabit dito sa phosphate group. Okay? The most common fatty acids that you will find in your phospholipids are palmitic acid and linoleic acid. Tama ba to? Wait lang. I should confirm that. Kasi may variety din kung ano yung fatty acids na nakalink dun sa glycerol. Hindi naman, not exactly pinaka-common yung palmitic and linoleic acid. But most commonly, you will find a carbon 16 or carbon 18 fatty acid at carbon 1. So, carbon 16 or carbon 18 and carbon 18 or carbon 20 at carbon 2 of glycerol. Gets ba yun? So, sa glycerol, kay carbon 3 may phosphate ka. Tapos, meron pang ibang functional group na nakakabit sa phosphate na to. Carbon 2 of glycerol, meron kang fatty acid. Ang common dyan is 18 carbon fatty acid or 20 carbon fatty acid. And then, sa carbon 1 ni glycerol, ang common na makikita ay 16 carbon fatty acid or 18 carbon fatty acid. So usually mas maikse yung nasa carbon 1 than the fatty acid at carbon 2. Okay. So the name of your phospholipid depends on the substituent attached to your phosphate. So kung simpleng H lang siya, meaning alcohol lang yan, that's your phosphatidic acid. Pero ang pinaka-common ay itong dalawa. Si phosphatidylethanolamin and phosphatidylcholine. So for phosphatidylethanolamine, so you have an ethanolamine attached to your phosphate group. And then you have a choline functional group attached to phosphate if it is phosphatidylcholine. So these two phospholipids comprise a big part of your cell membrane. So if you look at the composition of your cell membrane, you will see that probably 30%, around 30% for phosphatidylethanolamine and for phosphatidylcholine. So yung minor, you have phosphatidylserine, phosphatidylglycerol, and so on. So the important point here is that aside from this phosphate group bearing a charge, yung substituent na nakakabit sa phosphate group pwedeng may charge din, just like your phosphatidylethanolamine. So this enhances the polar head of your phospholipid. So mas mataas yung affinity niya with water kasi mas marami siyang charge, right? Okay, so that's for phospholipids. So again, ito, membrane ha. So yung function nito, membranes. Component of your cell membranes. Next, we have galactolipids and sulfolipids. This one, sa plants makikita. So these, majority of these are found in your thylakoid membranes. And ang difference nila from your phospholipid is that you have a sugar, a monosaccharide attached to carbon 3 of your glycerol. So may glycerol ka pa rin dyan. Di ba 3 carbon yung glycerol? So this is your glycerol. Ito yung 3 carbon. Yan. Carbon 1, 2, and 3. To carbon 3, you have carbohydrates. Kaya galactolipid. So for example, this one, galactose yung nakakabit sa carbon 3. And then again, you have fatty acids attached to carbons 1 and 2. So hindi siya limited dyan. Pwedeng disaccharide yung nakakabit. So for example, this one is digalactosyl diacylglycerol. So again, these are the ones found in the membranes of thylakoids in your plants. So question, bakit sa tingin nyo, instead na phospholipids, galactolipids and sulfolipids yung nakikita sa thylakoid membranes ng plants? Diba ang difference? Phospholipids, phosphate. Galactolipids and sulfolipids, carbohydrates. Bakit kaya? Bakit hindi na lang phospholipids din ang gamitin ng plants para sa thylakoid membranes nila? Any idea? Sino may idea?
Ma'am, kasi yung phospholipid po may charge, tapos parang given po sa thylakoid membranes, hindi po pwede. So, eto po mga galacto kasi wala po silang charge.
Okay. Good point. Pero, yung itong carbohydrates natin, marami silang OH groups. So kahit hindi sila charged, pwede pa rin silang mag-interact with the water. Right? So, in that aspect, pareho pa rin siya with the phosphates ng phospholipids. Any other ideas? Sirit na? So yung theory nila ay since yung phosphates konti lang sa soil, yung plant, so in the evolution of your plants, they learn to create these galactolipids and sulfolipids instead of phospholipids because their supply of phosphates, or phosphorus actually, yung phosphorus, their supply of phosphorus in the soil is very low. Kaya naging ganyan na carbohydrates yung ginamit nila. Since marami naman silang nagagawang carbohydrates, yan yung ginamit nila. Yung sulfolipids pala, ano lang siya, sulfated na carbohydrate. So at least one of these OH groups na replace yan ng sulfate. Yun yung sulfolipid. So same pa rin, carbohydrate siya, pero derivatized lang. Okay. Next, sphingolipids are another class or group, is another group of your structural lipids. These one are derivatives of your sphingosine molecule. So si sphingosine, so you see here the structure of sphingosine. If you notice, this part looks like glycerol, right? So, ito yung carbon 1, carbon 2, carbon 3. Ang pinagkaiba lang, si carbon 2, instead na OH yung nakakabit sa kanya, NH2. So, para siyang glycerol na meron ng fatty acid na nakakabit sa carbon 1. Although it's not a fatty acid, right? It's just a hydrocarbon chain. So, si sphingolipid, ang itsura niya is you have the sphingosine molecule here, and then you have a fatty acid bonded through an amide linkage. This is an amide functional group, right? A fatty acid bonded through amide linkage to carbon 2 of your sphingosine molecule. And then you have a substituent here. So kung H lang siya, which is yung original na nakakabit dito, that's what you call ceramide. Saan yun naririnig yung ceramide? Yung mga nakikita yung commercials. Saan yun naririnig yun? Ceramide complex. Tama ba sa mga shampoo? Di ako naniniwala doon. Di ako naniniwala doon. Anyway, so pag hydrogen siya, so the original sphingosine molecule, ceramide siya. So you have a class of sphingolipids called ceramides, and then depende kung ano yung fatty acid na nakakabit sa kanya. And then if phosphocholine is attached to this one, to this oxygen, that's what you call sphingomyelin. And this is the major component of your myelin sheath, that thing that protects your neurons, yung spinal cord natin. Okay? And then, it's also possible for this to be a sugar or a carbohydrate, glucose. So for that one, it's a general class of sphingolipids. A general class of sphingolipids called cerebroside. So kung glucose yung nakakabit sa kanya, that's a glucosyl cerebroside. Pwede ring disaccharide yung nakakabit sa kanya, tri or tetrasaccharides. For that case, we call that class of sphingolipids as globoside. And then, kung complex oligosaccharide na yung nakakabit dito, that's your ganglioside. Okay? So, these molecules, where do we see them primarily? Yung mga gangliosides, cerebrosides, globosides, etc. Ayun, sa neural tissues. Your cerebrosides are found in the plasma membranes of cells in your neural tissue. And gangliosides, gangliosides, what is noteworthy about your gangliosides is that they commonly contain sialic acid. Naalala niyo si sialic acid dun sa hexose derivatives na pinakita ko sa inyo. Nandun si sialic acid. So sialic acid, you often see them in gangliosides. And fun fact, yung nakadiscover nito, so syempre na-discover niya nung pinag-aaralan niya yung myelin sheath. Kaya niya tinawag na sphingolipid ay dahil hindi niya alam kung ano yung function nila. Dahil mysterious sila, kinuha niya yung pangalan from the Sphinx. Kaya sphingolipids yung tawag sa kanila. Ayan. So, fatty acids, triacylglycerols, waxes, and then structural lipids. So, under structural lipids, we had phospholipids, galactolipids and sulfolipids, and sphingolipids. So yung last class of fatty acids or lipids, last class of lipids ay sterols. So if you see your sterols like your cholesterol, sobrang ibang-iba yung structure niya sa lahat ng lipids na nakita natin so far. So far, nakita natin medyo magkakamukha sila, di ba? Basically, fatty acids, hydrocarbon chain. The rest, you have a base molecule like glycerol, kinabitan lang ng fatty acids. Magkakamukha sila. Pero si cholesterol or in general, yung sterols, iba yung structure nila. Okay? So, it's not a straight hydrocarbon chain. Rather, what you have is fused rings. Yun yung main feature ng sterols. So, the base structure is this one. We call it cyclopentanoperhydrophenanthine. You have 6 carbon-membered rings and 3 6 carbon-membered rings, yung ABC, and 1 5-membered ring, yung D. And they are fused together. Ito yung base ng structure ng sterols. Now, bakit sila under lipids? So if you look sideways in this 3-dimensional representation of cholesterol's structure, so may length siya, di ba? Pahaba siya. Yung length na to, kasing haba siya ng 16 carbon fatty acid. Okay? So, lengthwise, para siyang fatty acid. Okay? Ang pinagkaiba lang is itong fused rings. Now, your sterols are not limited to eukaryotes. Okay? Meron din, meron sa plants, like the stigmasterol. Meron sa fungi, like ergosterol. Bacteria that can produce sterols are very rare, but most bacteria can eat, so they can take in exogenous sterols and use it for their metabolism. So just like your phospholipids, sphingolipids, etc., cholesterol sterols, especially cholesterol, are mainly structural. So yun yung main function nila, part ng cell membrane. So the addition of cholesterol molecules to your membrane actually is a way for the cell to control the properties of the cell membrane. So kung mas marami kang cholesterol, for example, mas flexible yung cell membrane mo. But if you have less cholesterol molecules, your cell membrane will be more rigid. So nakokontrol yon ng cell. Aside from that, your sterols also have other functions. So some sterols can have fatty acids attached to them. So mas mahaba pa yung structure nila than your cholesterol. Some are part of your digestive system, like this cholic acid, which is a component of your bile acid. So, di ba, this helps in the digestion of fats from the food that you eat. And these are also precursors of your hormones. Yung mga steroid hormones natin, like your cortisol, which is a glucocorticoid. Of course, you have your testosterone, estrogen, which are your sex hormones. And then you have aldosterone, an example of a mineralocorticoid. So, anong ginagawa ng mga to? So, kapag glucocorticoid siya, this hormone helps in regulating the metabolism of your proteins, your lipids, and your carbohydrates. Aside from that, it also helps in inflammation, etc. Yun yung main function ng glucocorticoids. Si cortisol, ano nga ba yung si cortisol? If I'm not mistaken, si cortisol, it enhances storage of fats. Kasi si cortisol, pag mataas ang stress, mataas ang cortisol. Tapos ang tendency niya, for some people, halimbawa kakain ka, 'di ba? Since mataas yung cortisol niya, yung kinakain mo, madali siyang ma-store as fat. Yon. So more on metabolism yung glucocorticoids. Pag mineralocorticoid, ang kinokontrol niyang hormones na yan ay salt and water excretion. So more on the kidneys, the action of your kidneys. And of course, we are familiar with the role of your androgens and estrogens. Yon. So we've seen the functions of your lipids. Ano yung primarily nakita natin? Energy storage and insulation and structural part of your membranes, di ba? Aside from those, lipids can also be signaling molecules, cofactors, and light-absorbing pigments. So for example, we have what we call a group of molecules called eicosanoids. These are involved in the inflammatory response, pain, fever kapag meron kang sakit, pag may nilalabanan yung katawan mo. So these eicosanoids are grouped into 3, your prostaglandins, thromboxanes, and leukotrienes. All of these eicosanoids come from arachidonic acid, which is your 20-carbon fatty acid. So, i-derivatize mo lang yan. Pwede kang may prostaglandin, thromboxane, or leukotriene, and then your body sends signals to some areas where there is a need for an inflammatory response, for example. What else? Vitamin E is actually partly lipid. Ayan, because you have this hydrocarbon chain which resembles isoprene. Si isoprene is, this is actually the precursor of your cholesterol, yung isoprene. So kaya sinama yung vitamin E dito. Vitamin E is an antioxidant. So alam niyo kung ano yung ibig sabihin ng antioxidant, right? Na-discuss ko siya nung nag-discuss tayo ng glutathione sa amino acids. Ayan. And then lastly, you have some lipids that act as pigments. So they are light-absorbing molecules. Hence, they are the ones that cause colors. So for example, in the feathers of birds, you have these molecules that give the red color and the yellow color. Interestingly, the differences in the pigmentation between male and female birds are the result of differences in intestinal uptake and processing of carotenoids. So yung carotenoids, this is a class of compounds that can absorb light. It's because of the alternating double, single, double, single, double, single bonds. So because of these pattern, kaya nilang mag-absorb ng light and they can give off a certain color. Ayan. So that's it for the function and structure of your lipids. So pupunta na tayo sa lipid extraction. Again, it follows the same process. You choose your source, you homogenize your source, you extract your target biomolecule, and then you perform analysis. Now, syempre, ang unique sa lipid extraction is that you use organic solvents kasi doon soluble yung lipids natin. So your neutral lipids like your triacylglycerols, they are readily extracted by organic solvents such as these. But your membrane lipids, since they have a polar head, they are more effectively extracted by polar organic solvents such as ethanol or methanol. So, di ba dun sa procedure, kung nabasa niyo na yung procedure, first, you have the chloroform methanol mixture. Tama? Chloroform methanol mixture plus 1% sodium chloride. So, of course, the chloroform methanol mixture is for us to maximize the type of lipids that we can extract from the egg yolk. Kung chloroform lang, like what is said here, kung chloroform lang, most likely, triacylglycerols lang yung ma-isolate natin. So, kaya tayo naglagay ng methanol dun sa extraction solvent natin para makakuha din tayo ng mga phospholipids. Then, what do you think is the function of the 1% sodium chloride? Para sanang salt. Pag nag-purify tayo ng kahit anong biomolecule. Sa DNA, nasa exam niyo yon nung nag-exam kayo ng nucleic acids. Hindi daw naglagay ng salt. Anong effect? So, para saan yung sodium chloride? Anyone? Anong gusto nating matanggal?
Nagre-remove po ng proteins na pwedeng makakontaminate.
Yes. So kasi 'di ba, syempre especially yung membrane lipids natin. Since meron kang proteins na nasa membrane, yung mga embedded proteins. So gusto nating tanggalin 'yon. Yan yung purpose ng 1% sodium chloride. And then meron ding stage dun sa purification process na gumamit ng acetone. Right? Tapos, nung nilagyan ng acetone, dun ka na nagkaroon ng precipitate and supernatant. So, anong purpose ng acetone? Ano yung sineseparate natin? Ano yung nasa precipitate? Ano yung nasa supernatant when you added the acetone? What are we separating?
Ma'am, yung neutral lipids po ba yung nasa precipitate? Hindi ko po sure.
Wait, neutral lipids yung nasa precipitate. Di ba yung hindi ba phospholipids? Wait lang. Ala, nalito ako bigla tuloy. Nalito din ako. Ang acetone, ang piniprecipitate niya ay charged. Charged lipids. Tama ba? Tama ba? Wait lang. Hehe. Saan na? Mhm. Yeah. Charge. Yung phospholipids yung nasa precipitate. Sure na ako. Final answer. So, doon sa acetone step, yung nag-precipitate doon ay mga phospholipids. And then the rest, yung neutral lipids and cholesterol, nandun sa supernatant. Yes. I believe so. Okay. Yan. So, sa precipitate, phospholipids. And then sa supernatant, neutral, lipids, and cholesterol. Ayan. So, dapat makikita natin doon sa qualitative tests, magre-reflect dapat yung composition ng precipitate na na-isolate and yung supernatant. So, yung qualitative tests na na-mention, tatlo, right? Sudan-4 test, phosphate test, and Lieberman-Burchard test. So, Sudan-4 test is a general test for all lipids. So, wala siyang dinidistinguish na type ng lipid. So, this is Sudan-4. It's a very nonpolar molecule. And since it's a dye, it gives pigment if you have anything hydrophobic in your sample. So, kung meron kang, kung present yung lipids mo doon, dapat magko-color red yung sample mo. So, that's the positive test. Color red siya. And then, the Lieberman-Burchard test is to detect cholesterol. So, doon sa experiment, theoretically, dapat yung supernatant, positive siya sa Lieberman-Burchard kasi nga nandun yung cholesterol. So for this test, you have acetic anhydride and sulfuric acid. So what happens is that your cholesterol undergoes a series of dehydration, so natatanggalan siya ng water, and oxidation reactions. Kaya, meron kang series ng color change sa Lieberman-Burchard. So initially, your sample, if it contains cholesterol, it will be colored pink. And then, a hint of purple, kung mabagal, ma-observe pa yung purple. Pero yung final color niya is green. So it's because nagbabago yung electron bonding dun sa ring ng cholesterol. Ang ending niyan, magkakaroon siya ng alternate na double, single, double, single, double, single, double, single, double, single, double bond. Kaya nagkakaroon siya ng color. Ayan. And then the last is the test for phosphates, which was discussed in nucleic acids. Same steps. So kasi meron ka ring phosphates, 'di ba, sa phospholipids. So sa experiment, theoretically dapat ang precipitate magte-test siyang positive dito. Kasi nandun nga yung phospholipids. Kung effective yung pag-carry out nung isolation process. So again, yung test for phosphates, you have a yellow precipitate, which is your phosphate reacting with ammonium molybdate. And then finally, for lipids, you can perform thin layer chromatography. Actually, kahit ano naman, kahit sa, for example, sa proteins, pagkatapos mong i-hydrolyze yung proteins, pwede ka rin mag-thin layer chromatography para makita mo yung mga amino acids na nakuha mo after hydrolyzing the protein. The same, you can also perform thin layer chromatography for lipids. So, again, yung principle niya is that you have two phases, right? The mobile phase and the stationary phase. So the mobile phase is your solvent, the one that you place inside the chromatography chamber. And then your stationary phase is your silica plate. So on your silica plate, you place blots of your sample, diba? And then after drying the solvent out, you place it into your developed chamber. So pag sinabing developed, puno na ng vapors nung solvent yung chamber. So ilalagay mo lang siya sa loob, and then by capillary action, your mobile phase, the solvent, will rise up your silica plate. Now, the process of separation depends on polarity. So, pag simple or the basic thin layer chromatography, your stationary phase is polar. So, polar yung silica. And then your mobile phase is the non-polar phase. So, any molecule will choose to either stick to the stationary phase kung polar din siya, or it will choose to go along with the mobile phase if it is non-polar. So doon tayo nagkakaroon ng separation. Kasi kung yung mixture, yung sample mo, halo-halo siyang lipids, magsa-separate sila based on their relative polarities. Phospholipids will most likely travel a very short distance kasi mas mataas yung affinity nila sa polar silica. Cholesterol and your neutral lipids will most likely travel the farthest kasi non-polar sila and they have higher affinity with the solvent, which is your mobile phase. Just like your electrophoresis, you also run reference. Di ba sa electrophoresis, meron kang DNA ladder. Sa TLC, magra-run ka rin ng reference mo. So on the same plate, you place a blot of your sample, tapos magbablot ka rin ng, for example, pure cholesterol. So may isang spot ka ng pure cholesterol, may isang spot ka ng phosphatidylcholine, may isang spot ka ng triacylglycerol. Para mako-compare mo doon sa sample mo yung spots, mako-compare mo yung distance nila with your reference. Okay, kung sa agarose gel electrophoresis, we use ethidium bromide or some staining reagent to visualize the bands. In lipid TLC, ang ginagamit usually is iodine to visualize the spots of lipids on your TLC plate. So, ang ginagawa is, once na-develop na yung TLC plate natin, so tapos na, nag-run na yung solvent, nag-stop na, tinanggal mo na siya, na-dry na yung solvent, you place it in another chamber. This time, meron kang iodine crystal sa loob ng chamber. So, you have a chamber, you have iodine crystals inside. And then you have your TLC plate there. Tapos ilalagay mo lang siya sa hot plate. So parang mag-vaporize yung iodine. And what happens is that yung iodine, gaseous iodine, magre-react siya with your lipids. So kung nasan yung lipids doon, magkakaroon siya ng stain. Kasi nag-react yung iodine sa lipid na yon. So it's possible to visualize your lipid spots on your TLC plate. And then the same, you measure the distance, pero what we compute for is the RF, which is distance of spot over solvent front. So the solvent front is kung hanggang saan tumigil mag-travel yung solvent. That's your solvent front. So i-measure mo yan, and then you measure the distance traveled by the spot, get the ratio, that's your RF. And then you compare your RF with your sample, with the RF of your reference. So kung sino yung pinakamalapit, most likely that's your pwedeng phospholipid siya, pwedeng cholesterol. Yun. That's for thin layer chromatography. And that's it for this module. Ayan. Questions? Nabot tayo ng 1.5 hours. Okay. May questions ba? Wala. Gising pa kayo?
Wala naman na po ata, ma'am.
Okay.
Thank you po.
Sige, thank you. So, see you ulit next week. Bye-bye.
Bye-bye po. Thank you, ma'am.
Ingat.